any of you were able to progress in this further? But did you set up the pipeline?
So for this one, 2 data sets you're asking?
Yeah.
We have set up the pipeline, so it's 9% processed.
Excellent. So I
Yes.
think I'm, yeah. Okay, excellent. So that's, any other questions, any other queries and discussions we will finish before?
Aya is asking something.
I have question regarding raw sequencing from NCBI. Do to download for every sample as I had column? No, we don't. You don't have
Sir,
to download, no? Yeah, yeah.
Yes, sir. So can I help Aya after today's session? Once you are done, I like to sit with Aya and Zoran and help them prepare their files and run the pipeline.
Yeah, exactly. You don't need to download them. You just need to get the run table, no?
Yes, yes.
And make some modifications to the run table so that it is read properly in the pipeline. You don't have to download any GBs and GBs of data. The server is designed or is made to work with the NCBI repository directly, so you don't have to download this. So Sonalika is going to show you, and I will also show you how to look for what to do from this case, right? In one of some of the demo that I have prepared today. So any other questions? No questions right now. Okay. Anybody else? So let's go through. I mean, today it's like we're. It's not going to be. I mean, there are some slides if we want to discuss, but I think I will go through the process of identifying data set or selecting one data set, and then from a publication, an interesting publication, and then find where to get data from that publication, and proceed to download the data, and or at least prepare the RunInfo file from that data, and then set up DADA2 and QIIME analysis there, and proceed. That's what we will do. And yeah, let's start if we can. Let me set up some of the. Oh, what is that? Okay, wait a second. Yeah, let me set up the screen share. Okay. It's still working. My screen share is still not responding to me. Maybe I need to join and rejoin. Yeah, now it is working. Okay, great. Can you see my screen?
Yes.
Okay, this is an excellent paper that has a lot of potential, right? And I should actually thank Tanmoy, who is working with part of the data as a research fellow or research participant. And he's working with part of the data in this one. So he found out this data set, and this has a lot of information. And he's focusing on one part. We can focusing on a different part, or if you have ideas, we can also try focusing on other parts also, right? So let's go through what they actually did in this one, in this paper, is they their idea or their hypothesis of what they want to find out is participants who were given high fiber diet for a certain time period, and participants who were given fermented food diet for a certain time period, when they have measured the microbiome abundance or metagenomics data after 1 week, 5 week, 9 week, 13 weeks, and so on and so forth, do they see any changes in them in their microbial abundance using the metagenomics data. But they do not only get metagenomics data, characterize metagenomics data, they also characterize specific immune response also. And they also have a lot of other work to study the immune response for these people under different conditions. Look at this. That's what they actually did. There's a lot excellent paper, this one. So they studied the inflammation change, and they are correlating that inflammation change with the microbial diversity. So that's what they actually did. So we can follow some of the steps, or I can show you some of the association that I am trying to set up, and you can try to explore other possibilities in a data like this. One interesting factor that we want to discover or we want to find out for ourselves is they definitely observe this change in the baseline microbial diversity for people who take high fiber diet and for people who take high fermented food diet. So how much of this diversity or how much of this increase do we see? And at what time point? Because they also mentioned they have measured this at different time points. So at what time point do we see maximum difference in the microbial abundance? So those are the interesting questions. Some of the branched questions is, since they also characterized inflammation responses, right? So we can quickly take, irrespective of the food or fiber, irrespective of the diet they are under, can we annotate the participants into difference by their difference in the inflammatory response? So that has to be, we have to do that annotation, because they are not in there, they do not characterize the samples like that. So from this paper, if one wants to find out, you can find out. So then that can be added as another confounding factors. We can find out people with a difference in their inflammatory responses. How does microbial diversity change in them? So of course we have to group them by some group. Then you can use different methods to define groups in that, right? So let's quickly go through where to find data in this big paper, right? It's an interesting paper if you want to read. So it has lots of data. It has lots of data. I mean, yeah. I'll show you how much of data that this one has when we go and check their Bioproject. So this is a paper, and you can read through for some of their conclusions, right? So anybody who wants to work with microbiota and gut metagenomics and generally, so go to look for this Sonnenberg. He has an amazing publications. He has published since this one another 4 papers from this or from similar data sets. So all of his publications are really extremely worthy to be explored for understanding the research behind immune response and the microbiota and metagenomics data. So let's come down. While you see this, how to skim a paper. So you read the last paragraph of the introduction. So this is a summary, right? Come down, graphical abstract. We have this introduction. So you come down to the last paragraph of the introduction. Then they are going to say exactly what they have done. That's in the biomedical papers. All the biomedical papers go by this structure. The last paragraph is a very short summary of the whole paper. We read this, then we understand what they actually did. So what they actually did, so they try to address whether diets, right, that can be changed, that can change the microbiota, right, and whether we can use dietary intervention to change the immune status in normal healthy diets. So for that quantification or characterization, what they do, they study the effect of 2 diets. That's what is here, fermented and fiber-rich food. And this is part of the clinical trials. And anybody wants to check out the clinical trial information can go to this ID, right? And then they do not only this metagenomics profiling, they do other omics profiling also, but that's not part of our discussion here. But yeah, as I said, this study has a rich amount of data, right? So they also characterize immune profiling, and also what do they observe? They see that the fiber-rich or fermented food actually produces a distinct immune response. And for some of them, the responses are actually, as we would definitely observe, are general. For other people, they are very specific. So that's what they mention in the abstract, personalized response. So that's what I also discussed. So we have people for whom the diet actually induces a general response, which is like a baseline increase or decrease in their immune response. But for others, this diet produced very distinct signature in the immune response. So we want, that's not a comparison that I'm going to set up today, but if somebody is interested, this is an excellent data set for such exploration also. Okay, so what for how long did they study this? They studied over the course of 10 weeks. And 10 weeks. And also, what did they mainly find out? They found out that fermented food do modulate the microbiome immune axis, and they also give some way that it can be used as part of therapeutic suggestions also, or well-being suggestions also. So that's what they actually conclude. So skimming the paper, just read the last paragraph of an article's introduction. That will give you a lot of ideas whether do you want to pursue reading this paper or not. Okay, so you can go through the results. What do they find out generally? And we're not going to go through the results. And they see, for example, here, alpha diversity measures. They see some, let's do that. Click that, zoom in, yes. So what do they see here? Why cannot I zoom in? Oh, no. Okay, one more. Yes, good. Okay. So they find there is some, can you see this difference? They find there is some difference between in the diversity measure between fermented and fiber, and Shannon diversity also tells us, but I don't think that's going to be very significant. They don't see significant diversity, right? That's typically the case with majority of the human related data. An overall diversity measure is not going to be very significant for you. It's going to just show you which way the trend is going. So here we can see that the fiber diet has slightly increased abundance or measure, right? Abundance level compared to the fermented diet. And this is what I said, when you want to see the beta diversity tells you how different the samples are based on the grouping that we are defining. So based on the grouping that is defined here, fermented versus fiber, not much difference. Everything is clubbed together. So I don't see a lot of difference here. So they try to see whether the measurement during the course of these dietary changes, do they see, or are they any significant, or do they have any significance in them? It does not look like there is a lot of significance in there, but so they do see some changes in the diversity for different weeks. So these are all some of the things that you should note it down. So the analysis from here is not going to be straightforward when you try to or when you do or compare to the mouse data that you actually got introduced to in the last weeks, right? So this is something that we should keep in mind. And let's go down, and what else do they see? And now they are going to, for example, group it by different food intake, right? And if you want to group it by different food intake, and this is also some other phenotypical character. So that's why when I said this data is really rich, if you want to check the difference in the difference in their group intake, right? You can see how different categories changes. For example, at 14 or 10 weeks time, different participants have different intake. So we can actually select specific participant number based on their intake. If we want to see some of the participants for whom there is a very low vegetable intake, then I will select a specific participant, or very low legume intake. Then I select this participant for whom we do not see any legume intake at all, right? And very low nuts and seeds, a lot of grain in this participant. So you can still identify specific comparison here. So that's why this data set is amazing. So let's stop praising this data set for what it actually offers. So what we want to actually find out is these are proteomics data. Then we can come down to methods. You see that if you come down to methods, then you can definitely read how they perform this analysis. And they give some codes that they used for analysis in the GitHub. So you can go to this GitHub link and then access their code. And more data files for normalized output, right? For example, they also provide you normalized abundance estimation. So we can check that out also in this one. But our interesting link from here starts here. So they give the raw data, that is the amplicon sequencing data that has been submitted to NCBI and as under the project name PRNJ 743361. So if we go to this link, then we see that they have performed this analysis for example, 300 samples. That's a lot, right? So we can go to this SRA experiments. Then we have everything here, right? And we want to note down some of the things like Sonalika mentioned you, whether it's paired end or single end, or did you? I think she also showed you how to find out the read length of the reads for this. That's also there. 300, I think here. Yes, 141, I think, right? Yeah, 139, 141. That's a read length here. 2 reads per spot, so that's going to be 300. Is it 300 or 141, Sonalika? Are you here with us? We cannot find out from here.
So we should take the minimum read length here, so it should be 139.
139.
But,
okay.
Yes, sir.
Okay.
But then for all the samples, we have to take the average of the minimum. Average of the minimum.
Yeah. So that's that one we should definitely do. And another thing is, for example, they also should show me where for not for this one at least. If I take the run information, wait a second, where is this? There is a way to separate this by, for example, by the type of sequencing that they did. 16 GB, 10 GB, 9 GB. I am not so sure where this is. Yes, okay, shotgun sequencing. If you, oh, this is also WGS only. Okay, I actually, yeah, 2 into 1, yeah, 150 in this case. And where is the amplicon sequencing done here? And I'm trying to find out where the amplicon sequencing is actually done. How did I find out? Let's see, pardon. Okay. One way in which you can find out is you can look for this FIFO. That's what they actually mentioned. So, and this is also 1.3 GB. So this is also WGS only pattern. So whole genome sequencing only. Okay, that's okay. I remember discussing with him that this data set has amplicon sequencing information also. Where did we get that? Okay, let's find out by going through the run table. So we take the run selector and file, run information, and create file. And then we download this and we save it somewhere where we can retrieve this. Okay. Okay. My computer is really slow. Let me reshare one more time. It's very, very slow for me. So why would one use Shannon diversity matrix over Faith's phylogenetic diversity? Excellent question. So Shannon diversity, not only, for example, let me stop sharing and reshare one more time. Okay. Faiths give you phylogenetic diversity. Shannon diversity is actually related to, you can calculate, for example, species richness, how diverse each and every sample is, right? But you will get the Faith's phylogeny plot is going to give you, consider everything, mainly based on the difference or distance between samples that is obtained by different methods. Shannon and Chow diversity is going to give you how different samples are. Each one of these samples are based on the abundance, proportionate abundance levels each one of their samples actually has. So that is what we are actually interested in many times. So nothing against Faith. Different diversity measures give you different characteristics of the sample. That's what we have to take, right? And yeah, that's what we will also take. Wait a second, it's not opening up for me. So any other questions until then? Until I set up this screen share and we could discuss. We will go straight to setting up the pipeline now. My Zoom is really struggling. Okay, wait a second. Let me rejoin, maybe that will help. Okay, here. Why is this not working? Okay, I think now it will work. I just rejoined maybe. Can you see my screen? I think
Yes,
now it works. Yeah, okay. Now I also got some confirmation. Okay, let's not do anything with this one. Let's go back and then, for example, go back one more time. Yeah, and clear the. I'm just clearing the selection that I made here, and clear. Yes. So everything from 441, we have to download everything, I think, and it's difficult to make a selection here. So run information. If you create this file and open it in, let it run. Yeah, open it in here. Replace. Okay, let's go there. Okay, let's open this up. So this is huge. This data set is huge. It has everything. The whole genome sequencing is also done. And what we are interested in amplicon sequencing. So how do we find out? Maybe we can actually find this out from NCBI themselves, but better idea is, you see here, library strategy. In the run table, we can actually filter based on this strategy, right? Apply quick filter, and then you can use anything, numbers, or you can use this to it. Okay, now we have only amplicon sequencing, 311. Let's see how we can make this selection in the NCBI. Okay, single. Amplicon sequencing are done in single end, so we can use single end. So if you select that, it's going to give you only this amplicon sequencing data. And yeah, this is what I was actually interested in, the amplicon sequencing. In another way in which you can find out which ones are amplicon sequencing and which ones are whole genome sequencing are by size. You see, the size of the file is not in the order of tons and tons of GB. It's in the order of 100 bytes, right? Kilobytes, that's what. So that will also help you, right? Okay, so then if we go again, let's see, explore what other characteristics that we have. This is a very well labeled data. You see that, you can see that the sample names are here, right? If you come all the way down, for example, yeah, this name, right? This name has that specific characteristics. Like this is the name of the sample. We can sort by sample, then you will know this. Sort ascending or descending, whatever it is. Yes, okay. You see that this is sample 1, and this is after first week, after second week, after third week, fourth week, fifth week, sixth week, seventh week, 8 and 9. And that's how each and every sample goes. So you can take different time points and then get that specific data, and you can start quantifying them, right? And that's also very useful information. And you have this average length of the reads here. That's also given here usually, right? Many times this might not be given. So in that case, you have to go into some of them and then find it out. Because when we are going to set up this pipeline analysis, we are going to set it up only for a handful number of samples, like 10 or 12 at the maximum, not more than that, right? And for that, you can easily click on each one of them and then identify the read length from the NCBI page themselves. So this gives this information. How do I know that this after 1, 2, 3, 4, 5, 6, 7, 9? That is, this information is actually given in their method section of the paper. So if you go and read the method section of the paper, it's actually given there. So that's how we know. So now we can make some specific selections, like we can take sample for different diet conditions. And the diet conditions are not mentioned here, but again, the diet conditions are given in the associated resources, in the not in the, it's in the paper. So you go to the paper, and we know that which one of these 8,000, which one of these sample characteristics, right, or patient IDs, we're given what data, fiber data or fermented data. So we take that and then we modify this run table by adding 2 more columns, right? But we don't want to do it for everything. So I have done for some selection, and we will use that selection. For example, that is what is here, I believe. No, survey data. Okay, where is this? Yes. So this is what we will do. For example, we are going to select only these samples. Let's take this and open this up in where? In Numbers app. Yeah. Okay. So we take this, and then, for example, we selected only this data. How do we select this data? So this is selected, for example, based on the diet that they have and the time point. We want to, or I want to find out their progression of abundance level or proportionate abundance levels according to the after first week, after 5th week, and after 9th week. So this is how you set up this analysis is up to you, right? I am interested in this, and if you want to find out only after 1st and 9th, then it's fine. Or if you want to focus only on 1st week or after 9th week, so it's up to you. So how do you set up this analysis is that. So I want to follow the progression of abundance if there are any, and follow the difference in the abundance level if there are any, and if I can get hold of some relevant symbiotic and dysbiotic samples, and I can actually follow whether their abundance level increases after this due to the fiber or due to different time points after which this is measured. So this txt file, right, as discussed earlier, will go as a sample file or sample metadata, like how Sonalika described how to prepare that file, and she will also help you if you have such data. But that is useful for DADA2 pipeline. But for QIIME pipeline, we don't want to do that. There is no associated metadata file that you have to upload. So all the metadata information has to be modified, has to be included in the run information itself, right? You see all these different columns, right? Some of the columns will be used for finding patterns or associating or being a phenotype. We don't have anything here, so that's why I said we can use those specific samples. We have everything here, 300 here. We don't want to use 300 here, so use only for that, for these selected samples, and find first these selected samples, and then add these at the end. The next 2 columns should be we can make it as a diet and then time point the next. So after that, you can just save it as a .csv file, and then that can easily be read in. So this is how you can actually prepare these files for DADA2 and QIIME. So for what do we do for DADA2? We take this run information, and without any modification, we just select the samples that we are interested in and save that as .csv file. And then we prepare another file that is going to give you the annotation for these samples. Like I am interested in annotating these samples by their diet type, and also by the time point under which it is measured. So if you want to add like age or like sex or like BMI index and everything, it should go in the next few columns as a separate file, like metadata file or phenotypical data file. You can name that. So both these files will go in as input to DADA2 pipeline, and while all the interested columns should be added to the end of this big CSV file matrix for the QIIME2 pipeline, because QIIME2 takes only one input. So that's why we have to add everything in that run information themselves, which is going to give access to their phenotype information, phenotype there. So let's try to set up some of this there, and let's see if I can set up the pipeline there or find links for the pipeline. And where is this one actually? Okay. Okay, this is the pipeline, I believe. Let me reshare and share everything for you this time. Okay. Why is this not coming up? Okay. Wait a second. This is okay. Where is this file? Okay. Let me set it up for you. Let's see. I think this should work. I have to create this file also. Okay, let's do that. Oh, okay, so we will save this. Okay, let me reshare. Today is really bad. Okay, I just prepared one of the file for this, right? Save it as, and so, okay, can you see my screen? I just added these files so that we can set up this one. So I selected these samples, and then according to their diet and time point, I just saved this one. So we just export this file as .csv file. I export to CSV, and then export to CSV. Next. Okay, we have to go to this one. Okay, run table total. Okay, for this is for QIIME. Okay, let's do that, and then we have the other file ready, and that one is here. SRA data is actually here, and this is going to save as export to. We can just copy it and paste it. Don't have to save it there. Okay, this is SRA data is here, and that is okay. We got it. Okay, so this is what you will get, and let's set up some pipelines. And let's first set up DADA2 pipeline and see what are the conditions or what are the considerations that we have to make for this, right? Okay. Okay, so you go to amplicon sequencing. So that's a DADA2 pipeline. So we first have to, let's see, we say that this is a FASTQ file, and does it make any difference? Doesn't make any difference, yeah. And the type of databases is local databases. So earlier we used to have, yeah, we use select all, and then we go to file upload, and we upload the files from here. Since we are going to upload run table information, so then we go to this specific run table information, and then we upload this file. For this is for this, but we can upload this also because the selected files are here. Then we go to supplementary text file that I have to upload, and I upload this text file that I just prepared, sra.txt. So this one. Okay, so we have this, and then submit, and we start. What do we do? Right. So we starting this doesn't do anything, perform some quality checking, quality control, and everything, and then we can filter and trim using Trim-O-Matic and filtering. So this is very useful, for example, to examine the quality of the reads, right? We can trim this, and do we have to change the minimum length here, Sonalika? That's not here, no?
Yes, sir. No, sir, not here, sir, but
Yeah, that's okay, yeah. Okay, in the filtering, I'll show you. I think we discussed this also. Yeah, here is where I have to change the read length, right? Forward read length and reverse read length. Yeah, I got confused. So this is one.
so also, is it important to select Trim-O-Matic before QC filtering?
And not much. We don't have adapter sequences. We don't have to select Trim-O-Matic actually, right? Maybe, yeah, maybe we can actually go back on this. I got, I usually do this, but yeah. So this is forward read length and reverse read length is going to be 150 and 150 because 301 is what is given there. It's actually 300, and you divide the 301 by 2, it gives 150 point something. So you choose 150. Okay, and what you do, and then we save it, and then we go to replication count. But before that, I want to actually show you this is going to, this quality control is going to give you some important plots. Once you have your analysis done, it's going to give you plots like this, right? So after some of your associated output files will have these read quality plots. So this is what you have to first check. In the read quality plots, you see on the left, I have good quality reads. On the right, I have really poor quality reads. And why is it very extremely necessary to do that? Because good quality reads, and they can make or break your data analysis, right? So if you see any of the samples with extremely poor quality, you can eliminate that sample and then choose a different sample if you have the luxury of doing that, or you can actually completely eliminate that sample from the downstream analysis part. So how to eliminate that samples? That's going to be pretty easy. You can just remove that column. So that's why it's good to start with more number of samples. And if you find some problematic samples, you can just leave out everything. But don't take 50 or 60, finding related information and finding pattern is going to be difficult there also. So another plot you will see is this kind of plot, where it's going to show you what a good read length is, and for example, how to avoid, for example, how to find problems in your files here, right? What you see are the error rates that are actually observed for each and every consensus quality score from your file, right? And the black line that actually shows the estimated error rates after the analysis, and while the red line will actually shows you under proper definition of under normal conditions, what is the error rate that you should expect, right? So if the black lines actually come and fit very well with the red lines, we can believe those data a lot. And if the black line is completely pathetic or poor for completely different from red, then we cannot believe that data, right? Okay, so what is this? What does this actually tell us? It's checked for example, possible error rates, right? For example, if we have G's, or what does it say is, if you have this is for one file, you'll get a plot like this, right? This is generally good error rates, except for a couple of places here and there. So that's generally what you will see there. If you see the trend that is followed, that's good. This is excellent file. This is not so excellent at many places, because the red and blacks are very almost identical here. But here we have some problems in this, in a couple of transitions here. So when you have reads that involves guanines or thiamines, then we have some problems here. So this is not great sample, and this is excellent sample. So that's what you should keep in mind when you are performing this analysis. So let's go back to this one and set up this analysis. So that's what this will actually give you also. And then we can go to replication count, or replicating the sequences, where it actually count the number of OTUs in your files, and then it is going to, for example, it's going to reduce the computation time, for example, when you have redundant comparison. But what does it compare? It compares the unique sequences, right? It combines all the identical sequences that you will find, and then combine them into these OTU abundances, and then we can just simply count the OTUs and estimate the abundance for OTUs. So these OTUs, how does it identify the identical sequences? These OTUs are defined by the number of, for example, identity levels, right? Generally, OTUs are defined at 97% to 99%. So when you have these OTUs defined at 99%, you are mentioning that the sequences that are extremely close together, I consider them come from the same species of bacteria, right, or microbe. And if you have a very varied or very variable sample, and if you think that the sample is going to be extremely variable, then you can relax this 99 to 97%, this one. So generally this is fixed. Now you cannot change this in DADA2, but generally this is fixed. So at 97 to 99%, so that is there. And you can move further to, for example, cluster these OTUs into those specific OTU clusters, right? And identify as one the sequence that is going to characterize each one of these OTUs. These OTUs has to be defined, no? They have to be, for example, defined for their sequences. We can define them like OTU1 will have these blue sequences that is characterized by presence of A in this position, and similarly for this OTU is characterized by the presence of G in here, and this is by T in here. So this will not be as simple as that. They'll have multiple positions that are unique. So that is what clustering actually helps us achieve, right? And then we have to remove the chimers. And what does remove chimer mean? Is actually removes chimeric sequences, which involves 2 or 3, for example, reads from 2 or more bacterial components, right? We here, for example, when we are when we are actually preparing the library, we take utmost step to make sure that maintain that the samples that we observe are specific to one type of bacterial component or one type of microbe. Sometimes what happens is when they are sheared and when they during the process, you will observe chimers where part of the sequence is from one microbe and the other part is from the other microbe. So that is not going to be helpful for us to perform analysis. So DADA2 can actually remove these fuzzy OTUs or OTUs that does not combine into any members in the clusters. So they are removed for their chimeras, right? And then once you do this, then most of your noise is going to be reduced. So now we are going to have only OTUs that can be characterized by their specific sequences and by their specific identity levels. And now we can use these OTU sequences to find out from which microbe or bacteria is that from. So this is usually done by comparing it with the Silva database, mainly Silva database. And there are some options to provide additional databases locally, additional defined databases. In here also you can do that, but for 99% of the purposes, this is going to be sufficient for characterization. Silva is going to be sufficiently suitable for characterization. You remember that we selected all known databases, and that includes Silva option, right? So that's where the OTU sequences will be compared to, and their taxonomic identification will be performed by comparing their 16S sequences to the database, the repository. So we don't have to perform the reference sequence. This will become the reference sequence, and this is where you perform this reference sequence. And then we will, until now, we have all the information that is necessary, for example, to after assigning taxonomy, it is necessary for performing some abundance calculation, right? But for some of the ordination and abundance calculation, we see that it is common to create phylogenetic tree of the taxonomy and then use that phylogenetic tree to for metrics like unifrac distances and faith reconstruction, right? And these methods, right? To calculate beta diversity. So for that, we use a neighbor joining method to create a phylogenetic tree of your OTUs, and we save this, and then that information is what is going to be given to PhyloSeq, which will produce all these different abundance plots. Here you can give a selected taxonomy levels selection. So this is going to affect only, for example, let's see your output, the PDF output that you get from the pipeline. This is going to affect only that, but you will obviously have this option to change it in the external dashboard, right? That's what we will see. And data visualization, we select this option for dashboard and other visualization option, and then we end the pipeline here. And while ending the pipeline, you give it a name and end the pipeline. So we that's how we set up the DADA2 pipeline. And I'm not going to do this. Let's reset and then go back to how to get and how to analyze the output from DADA2 pipeline, which we already covered a little bit, right? And it's, if you go down, right, and we mentioned this, we covered this a little bit in the human diet and chow. We covered a lot in this one. And do we have the link for this, or do I have to go through all over this again? I think you know this, right? We covered all of this, what to look for and how to read the output, right? And I will quickly open this up and show you where the error plots are that we did not get access to in the previous sessions. And then we will use the same input and set up a QIIME analysis. And I think we are already running late. I think we should have another 15 more minutes. Is it going to be fine for you? Is it good for you? 15 more minutes? Am I still audible?
Yes, sir, you're audible.
Yeah, I think, okay. I see chat. Oh, I'm sorry. I think I did not open up the chat window. Let's take some questions here. So what do we have? To set up the read length, did you add the read length FNR divided by 2? Yeah. Yes, actually, yeah. If you are going to, where is the chat box? Yes, okay. So if you go by identifying read length from NCBI web page, then you have to add F plus R by 2, right? That's what you do. Or if you are going to get the read length by your SRA file, if it's there, you observe here, the read length that is given here is a combined read length. Not this one, the other SRA data. Okay, no. Run table total dot numbers. Yes. If you see here, where do I see that? Yeah, here average read length is this is a combined read length. So so if it is, because how do I say it is combined read length? Because we have only one file or one link for one file, right? This is, yeah. So that's what we take, and yeah, if it's forward plus reverse, it's 300 by 2. That's what we do. So let's go back here. And while setting up, let's, okay, what else? DADA2 cluster the sequence based on ASV, not OTUs. No, DADA2 does it by OTUs. ASV is for QIIME, right? But people use this interchangeably, right? Because ASV has a different way of coming to the amplicon sequence variants. DADA2, if you call that by OTU or ASV, the background process is taking the or finding related clusters only by their sequence identity level. Let's find out difference between OTU and ASV here. Do we have that? Do I have that plot somewhere mentioned here? Where is that? I don't have it here. Oh my goodness. Okay. Okay, I don't have it here, but what it actually does is in one of the other keynote, I actually prepared a plot like that for OTU and AS, comparing OTU and ASV. So you take only the sequences and identify the similarity levels and then cluster them. That gives you OTUs. But when you cluster them, you look for probability of these OTUs to be together, and based on that probability, you create distances between OTUs, and you use that distances and the sequence level to actually define which ones are which one forms different clusters. That is your ASV in QIIME2. You don't have to do that, but the algorithm does. So the algorithm actually creates a probability of two sequences or two specific reads to be not reads, right? Yeah, two specific reads to be with each other. After we run DADA2, the clusters, then we recluster based on no. If we are going to use QIIME, this is actually, you will see that when we are going to discuss QIIME's pipeline, I'll tell you where it's actually defined ASV. We take the reads, we define ASV quickly, and then we use that ASV, and then we run DADA2 inside QIIME. QIIME actually uses DADA2's algorithm, but before submitting the data to DADA2, it runs generates these ASVs by a probabilistic based method. So what this probability method, which actually estimates not only the sequence identity, but based on the sequence identity, the probability of two reads to be closely related. So based on this probability, you will find some of the reads that does not fit anywhere, and these are eliminated completely in QIIME. Let's yeah, that's where it's kind of. Let's find, for example, yeah. So you can see my screen, right? I will show you when we are dealing with QIIME, when we are going to deal with QIIME in short while. So in this output file, you can see the error plots here. See this error plots? This is where you will get all the error plots that I showed you. So even before you are going to go through your analysis, you just take a quick peek into these error plots. It's not absolutely, most of the data is going to be fine. So I had to really nitpick to get some errors here. So just take a quick peek into these error plots, then that's going, most of them, then it's going to be good. And we discussed the dashboard really extensively, and how to get data from the dashboard, and how to use that for collab, and that's also discussed extensively. So let me set up the QIIME and then come back to you, and let me also find that specific graphic. That means if you use QIIME, you will run the sequences exactly something like that. Yes, and that's not only that, and I will show you some other advantages of using QIIME also in short while. Let's see, where have I, okay, it's where have I prepared the file specific, where is this, where did I discuss this? Is it here, maybe here? Okay, maybe in this one. I'm just looking for just only one slide. I'm not able to find that one particular slide where I discuss difference between DADA2 and QIIME in terms of OSVs and OTUs and ASVs. I think it's this. Okay, I mean, just give me two more, couple of more minutes. If I can find it, then I'll find it. If not, then I will discuss this later. And where else do I have this? Okay. It's not here. Let's quickly check one more time. I think this is the latest, this is the file that I prepared last. Where did it go? I think this has to be in this one. Yes, okay, got it. Let me find this. Where is the zoom? So we have to quickly go to this also. So for example, so yeah, so you can see that OTU just merges your sequences by their identity percentage, right? It doesn't calculate or it doesn't estimate for whether or not that identity percentage might have occurred by chance or by anything else, right? But what QIIME actually does is, after finding out related clusters, right, then it actually removes some of the other this also. Identifies these representative sequences, and based on these representative sequences, then it will actually look for clusters that is going to define those two, come close to those two reference sequences. So that's what actually QIIME does. So OTU just clusters and then merges everything together. But QIIME, depending upon ASV, depending upon your percentages and depending upon your threshold for removing some of the noise, then it actually removes those noises, right? That's what it actually does. Okay, this, okay, let's see, where else do I have this QIIME set up? Yeah. Okay, so let's go through, where is this? now. Yeah, so it's about the probability of those sequences being together within that identity percentage. So that is okay, not this one. Why is my Zoom not finding my exactly? Yeah, it removes sequences that do not belong to any assigned clusters. Also, it removes sequences based on the probability percentage, right? The estimation of this probability is what is important for QIIME. ASV, right? ASV removes a lot of noise also. That's why it is kind of preferred if you have data that is very varied, or if you have data, for example, that is from mouse data, or data that you can see my screen, right? Because I'm not getting confirmation that you can see my screen at all. So it says I cannot share the screen. I cannot share the screen for some reasons. So if you can see the screen, that's fine for me. Yeah, irrespective of whether I get confirmation or not. So merging the sequences by their identity percentage, be it whatever it is, 97 or 99, that is OTU. But finding the sequences probability of being 97% close to any other sequences, and if this probability estimation is not going to be after the threshold, then those sequences are completely left out, and they are considered as noise sequences, or at least noise, and hence you have amplicon sequence variance, ASVs, and you will get a fewer number of sequences participating in ASVs, but those ASVs will be very well defined. You have, for example, OTUs, you have the chance of clubbing two strains or two classes of bacteria or two species of bacteria that show extraordinarily high sequence identity close to each other. But in ASV, you eliminate that chance a little bit. And QIIME, for example, if you have a same species of bacteria with two different strains, and that show some sequence variance that is greater than 97%, then you have to consider them as separate. And that again, QIIME actually takes care of that also. ASV actually takes care of that also. So for QIIME, let's open this up and let's set up some analysis. Let's see how do we want to upload this manually, or this one, the file is not multiplexed. Right? Do we have to change this? Yes, it is multiplexed. Let's see. I believe that is one. Manual upload, NCBI upload. What is these two? I think it's NCBI upload. Yes, okay. NCBI upload goes to NCBI run table upload, which we have actually prepared for that. So let's go take this, and then this is what I will upload, and go next. And here is where it gets really interesting, right? For some of them, right, you can define custom region, but if you can find out their amplicon sequencing data, let's try to find out which region was actually sequenced for the file that we are considering. So the file under study is not this one. Let's go to this SRA data. So the file under study is this one, SRR 8424. So let's go back to this place, and let's find that SRR 8424. Where is this? I think this is, yeah, this is 311. This is amplicon sequencing only. Yeah, it says 16S amplicon sequencing. And you see here, V4. Can you see this? Can everybody see this? V4 region is what is being sequenced. If you go inside, I think this also will show you which region has been sequenced. It's not shown here. I think we might have made a mistake in this one. It's okay. Let's see. Yeah, V4 is what is sequenced. So this is what we have to find out, right? You can check for others also. Let's randomly check for the other one. And again, we see V4, and V4 region is what is sequenced here. That's the information that we need to set up this one. So go and select V4, and that's going to take care of your sequences. So submit, and you start. And yeah, let's load up. Let's say okay. And then we import data into QIIME's environment, which is what QIIME actually gives us, right? Okay, it's not selected now. Oh, I thought, okay, that is fine. And then it gives you lot of, for example, freedom and flexibility to do this, right? When you use QIIME's environment, I'll show you what is those freedom and flexibility, right? And yeah, and let's go next. Just leave this in minimum number of reads in sample. Let's be 100. Let's leave this, and then go to quality control. And QIIME also has a way of checking the quality control, and you will see the outputs also. So you see that it's going to use DADA2 inside that also, processing OTUs. But the way that QIIME is actually going to work is it's going to create or define ASVs first, and then is going to use DADA2 to use DADA2 on the ASV that they have actually, or QIIME has actually worked on, right? And then we perform Silva classification based on the sequences, right? And then we go to, for example, remove unwanted taxa. So if you want to remove specifically some of the taxa, you can mention here. So we are going to deal with prokaryotic sample microbes. So here it is given that if by mistake, if I have any eukaryotic samples or mammalian samples, then remove that. So that's what is given here. And if you want to include eukaryotes and mammalia here, right, you can include that also to perform some of the analysis, right? That's also that you can, if you want to include, you can include that. And again, as I mentioned, phylogeny is a method with which QIIME creates lots of this one. No, QIIME will take care of that. Low quality sequences, QIIME will actually take care of that. We don't, most of the time, you will not experience trimming sequences at X base pairs. Why? Because the amount of, when in amplicon sequencing, is very cheap, and people go for high depth, enormous depth in amplicon sequencing, and then you don't get a lot of differences in, for example, you don't get a lot of bad quality reads here at all, and you have enough number of reads where these bad quality reads will be eliminated, or that position will be eliminated by being a technical noise, right? And the whole 16S genome is 1200 nucleotides or bases long. That's that simple. And if you're going to look for some specific hypervariable regions like V4 or V1, V2, V4, some combinations of this, that combination is going to come to less than 300 base pair. So you will not have to trim some number of base pairs before and after. So that's why Trimmomatic is not used in either QIIME or DADA2. So I selected that by mistake because I've been setting up some RNA-seq pipelines, and it came by habit. So let's, yeah, then by core metrics, it's in this data you're using the quality of reads drop as you go along the sequences. No, the quality of reads don't drop here also. We will either have good read or bad read, because the number of reads are going to be very small, in the order of 1,150, something in that length only, right? So you either have good ones or bad ones. Only when you have up 300, 350 long reads, then you will have some positions that are good or bad, and that's also that also has to deal with the coverage also, right? The coverage also defines the quality of the reads. If you can perform enough extraordinarily high coverage sequencing for transcriptomics data, then you will not see a drop in the quality base calling quality in the read end. So that I have seen that also. In some extremely high quality transcriptomics data or exome sequencing data, you will not find drop in the quality of the reads. So you will not find drop in quality of the reads here also. So yeah, we can mention sequence richness and sample richness and diversity here, and then we can obviously create some rarefaction plots and diversity visualization. We choose them. We by now know that what is alpha and what is beta diversity. So alpha is within sample diversity and beta is between sample group diversity. This is if it happens to you, this happened to me after a long time. So if this one happens to you, like save it and interactive visualization, just remove the beta diversity visualization. You can right click to remove the remove selected option, right? That we did not tell you. So I have selected alpha diversity. If I have to go back one step, you right click on this, it says you want to jump to previous step. Yes, okay. So I select again alpha diversity, and then I don't select beta diversity and go to interactive visualization, and that's going to select okay. This is happening because we went through this visualization option and we don't have the option to visualize beta diversity. That's why beta diversity visualization is giving you some trouble. And let's say it's okay, and then give it a name and give the pipeline a name, and yeah, run the pipeline. I'm not going to run it now, and I will show you one of the pipelines that I actually ran. Okay. For some reasons, this is also going to okay. I did not select that. That's why. Okay, how can I go back? Unfortunately, I cannot go back. Okay. Yes, interactive visualization. If you choose, and this comes up, I forgot. So this is like this is where you have to set up what are the phenotypes that you are going to visualize. Do you remember this release date? Where is this release date coming from? If we select this is a run information that we actually supplemented to QIIME, right? It's here, right? This is actually selecting the first column. You see that release date, it's actually selecting the first column in this one. That's why I asked you to add diet and time point or the phenotypical characters that you want to find, study their association and color by coloring the samples in the last two columns. So that is what we will use here. Let's select first, go all the way down to select diet, and then select 2, go all the way down to select time point. And we submit, and then you can end the pipeline. And we can end it, save it, and let it. Okay, I don't want to do this, and then I already have this pipeline done, and that is not here. QIIME2, yes, it's actually here. Okay, this is what this one is, how it is done. Sometimes the beta diversity will work. I think it's here it worked. What do you observe here? Here is in QIIME, it is actually give you a lot of QEZV and QEZA kind of files. And QEZV is a file that you can visualize into QIIME Viewer. And QEZA is a file that is that are data files for these visualization pattern. That is called A is for artifacts, but that actually has exactly, yeah, A is for artifacts. But if you open that file, you will find tables organized in a way that QIIME reader can actually read it. That's why it is A. I actually forgot what A meant for. So V is for visualization, A is for artifact. So you can download this, all Q is A and this one, and also all Q this one. Download this and visualize this in For example, in a QIIME environment, what is a QIIME environment? If you go to QIIME visualization, you can use this to visualize. Why do we have to use this to visualize? Because the external dashboard and this one and tax abundance plot is not going to give you all the parameters to visualize. And that part we are still building up, at least until we finish building this up, then we have to use some of the QIIME associated files. So let's download this quickly. I think I have done this, but I forgot where I kept it. Save this quickly. And hopefully it's 250 megabytes. Okay. And let's try to give some moral support by pushing this. And let's see if I have this somewhere. No, I don't have this. Yes. Okay, let's finish for this to complete. It's almost there. So we have discussed with the external dashboard extensively, right? Because I don't want to go there again. And you all know how to navigate the external dashboard and find relevant information. So let's let the QIIME2 all the QIZVs and this one finish. It is done. So let's go there and then extract this file. And where did I keep that file? Here it is. And look at this. We have, can you see this? We have, or can you read this? What happened here? Yes, Sonalika, I know that it's almost done. Wait a second, maybe 5 more minutes. So a lot of QIZD files and QIZV files, and we want to focus on QIZV files. How do we want to do that? We go here in the QIIME viewer, and if you drag and drop this QIZV file, it's going to show you. It's going to show you the visualization. Let's go here. For example, the Bray-Curtis visualization, if you drag and drop this, is going to show you here the visualization for our sample that we actually did. You can explore this. You can select color for category. So this here also, you're going to have all the columns from your SRA table populated here. But here it's going to be organized in alphabetical order. So our interest is in diet. It's here. Fermented fiber is in this place. And okay, I see that it's not differentiated by diet. Maybe I select some other time point, some other annotation like time point. Let's go to time point. And there is some correlation here. You see that majority of the ones are focused here. And except for some of the 5s, the 5s and 9s are 5-week and 9-week measurements are far away from the 1 from this plot, right? That is what we actually give from the beta diversity plot obtained using Bray-Curtis estimation or Bray-Curtis. So you can explore this. You don't have to color this by this one only. You can give shapes, right? You can select diet for the shape, and then you can give these, for example, you can select this as ring. So rings are fermented and these ones are fiber. So you can change everything. You can go to axis and then change the background color, and you can do everything. So you can do the customization here, and we know that how to do the customization. So this is good. And then let's select some of 1 or 2 more, for example, visualization option, like somebody was interested in faith group visualization, and faith group is going to give you options like this using phylogenetic tree analysis. So here also you have to select this, right? And I want to say diet, and this gives you grouping information. So diet, this is alpha diversity, but estimated by faith group as one of the parameters. And if you scroll down, you will see the p-value here, right? You see the p-value here. That's very important. This is very close to being significant. 0.05 is very close to being significant. So I see that for the selected samples, we see a very close to being significant difference between fermented and fiber diet change, right? And yeah, and let's go to some other information. You can download the files here, right? Download this as raw data as TSV and download this and for further customization. And let's take one more. And what are you interested in? For example, let's see sample bar plots, and this one is going to just give you okay, based on this level. Okay, this we already have. We don't have to go to this relative frequency to here, but we can use the output from our dashboard. And yeah, here, weighted unifrac distance. That's what I was more interested in. Let's see what is this alpha rarefaction also that we can check quickly. Yeah, so it's good, the sequencing depth, and this is really good, and you can explore this a little bit more further. And we will quickly see the beta diversity is weighted unifraction, because beta diversity plots are the one that are not given. What is the default threshold for p-value in QIIME? It's 0.05. That is what is default threshold is, weighted unifrac emperorship. So okay, that's a bummer, because I don't see any sample differentiation by weighted unifrac here. Maybe unweighted unifrac is going to be more informative. But do I do unweighted unifrac? I don't know. Yeah, so let's check. Unweighted unifrac is here. Let's check unweighted unifrac. Sometimes unweighted unifrac is better than the weighted ones. And body site significance. Okay, let's check this one first. Okay, there is some sort of change. Again, diet is not very informative in separating, at least between samples, right? Okay, time points also a little bit there, right? Again, the same thing. We see, for example, this is mostly 9, and yeah, mostly 9, but there is some separation, but that's not very good. But this is 3D plot, so you can check the 3D plots on their visualization. Okay, I think that's what I wanted to show you. You can explore all possible options here, and then you can make good pictures from this QIIME for beta visualization and this also. So what will happen if you take this A file and then put it here? That's going to show you, for example, what format is this one like? Yeah. Oh no. Oh, this has only details of this one. Okay, I thought A file is going to give you information about the table. Why does not that give me information about the table? I don't know. Okay, let's check this one, PCOA results. Why? Okay, that's really bad. Yeah, I know why. I know why. I should not open A file here, right? It's actually, for example, for the A file, you have to extract using some extraction method, right? Let's check this one. I can, okay, for the PCOA results, let's open with archive utility if you have Mac, and then that is going to open up here, like you have data, and you have data table here. That's eigenvalues. All the eigenvalues for each species is given here. So that you can use, but we hardly need it, because whatever is here is going to be plotted in the in these ones here. So for sometimes we might need to get yeah, we might need to get the table, and I'm not so sure if it's, yeah, it's okay. This says you can view Q is a day here, but it's not helping me to view Q's a days, right? Detailed provenance. Yeah, for some reasons it's not giving me that. I'm not so sure. I just have to check. I don't know for which A file that I found this one coming out. Let's check one more, and then we will finish. And the distance matrix. Let's find some matrix. Okay, breakout is distance matrix. I am interested in OTU's taxonomy. Again, it's not giving me. Okay, that's, if I find out, I will let you know. But we are not going to deal with A, because the abundance table, we only need 3 tables, and all these 3 tables can be accessed from the external dashboard. So that's why you don't have to use the artifacts file from QIIME Viewer at all. QIIME Viewer is for viewing some of the beta diversity and these plots that as of now you don't get these visualization option from external dashboard. So future we will build it up, but until then you can use the QIIME Viewer to visualize this. I think this has become a monstrous session. I will stop by now. I hope all of you would have gotten comfortable in setting up DADA2 and QIIME pipeline, right? You can choose either of these depending upon your data set, and that is going to help you also. If you have any questions, please, we have something like 3 or 5 minutes to go through some of your questions. Do you have any questions until now? Questions?
Sir, I think we can take up the questions via
Later?
email. Yes, sir.
Yeah. We are late for another meeting. Sorry about that. Okay.
Yes, sir.
Thank you so
Thank
much.
you, sir.
And good luck with your analysis, and do shoot us an email if you are stuck somewhere. We will discuss. Thank you.